Welcome to Module 4. We will cover this week the evolution of genes through the study of population genetics. In Chapter 2, we looked at the history of scientific thought that led to Darwin's theory of evolution by means of natural selection. In Chapter 3, we examined the inner workings of the cell and how DNA replicates itself and codes for making proteins. It is time to merge these grand ideas. If natural selection leads to the survival and reproduction of individuals with particular phenotypes best fitted for an environment, and if these phenotypes are caused by underlying differences in the genes, then evolution can be studied by looking at the gene frequencies themselves. If these gene frequencies change, then evolution must be occurring. This approach, called population genetics, examines how gene frequencies vary from one population to another and how gene frequencies change over time. The big question we will address in this chapter is, what causes evolutionary, and by what we mean genetic, change? For example, how did changes in genes and natural selection combine to form organisms so amazingly adapted for the environment, as this leafy sea dragon? This is not a class on leafy sea dragons, so we will use many examples from humans as well. In the previous chapter, we focused on how genes move from the parent to the offspring. In this chapter, we will be talking about groups of organisms exchanging genes. Let's start with some basic terms. A group of organisms that interbreed and have offspring is called a deme. If you combine all of the genetic information in an interbreeding population into one large group, we refer to that as the gene pool. When a group of organisms cannot interbreed with another group of organisms, or if they do interbreed but they produce sterile offspring, we often will group those organisms into different species. The species concept is a bit messier than that, but for now, let's just consider species to be groups that are reproductively isolated, meaning they cannot or will not breed together. Dogs and cats, for example, or gorillas and chimpanzees, or giraffes and deer, or ostriches and emus. The image on this slide shows the closely related ostrich in Africa and the emu in Australia. Though they share a common ancestor, they no longer breed with one another, in part because they are separated by considerable distances. This is an example of how reproductive isolation can sometimes happen because two populations simply do not encounter one another in the wild due to some geographic barrier, such as a mountain range separating these two species or a large body of water. This can, over time, result in the accumulation of enough genetic differences that even if the two populations become reunited, they would not recognize one another as mates. This is one of the major factors in causing new species to evolve. In later chapters, we will be looking at species of ancient primates, apes, and humans. Meanwhile, if all we have are fossils, how do we know that these populations were reproductively isolated from one another and were, in fact, different species? We have to rely on the assumption that if two fossils have similar enough phenotypes, like tooth morphology, they must have been sharing genes and therefore could interbreed. If there are substantial differences in anatomy, perhaps they were reproductively isolated and represent different species. It is sometimes quite easy to determine from the fossil record which modern species the fossil most resembles, as is shown here in the case of horseshoe crabs, cockroaches, and opossums. These living fossils closely resemble the modern form, demonstrating that natural selection has favored these anatomies over a long period of time. However, many fossils look quite different from their modern descendants, making it difficult to discern relationships. Complicating matters even further is the fact that many fossils represent dead-end lineages that did not leave any modern descendants. Sorting this out is difficult and will be looked at again in later chapters. By studying how gene frequencies change over time, we can examine small-scale changes over a few generations, something called microevolution. This allows researchers to very closely document genetic change and to seek out the causes of this change. For example, in this species of the beetle shown at the top, the frequencies of the green allele changed from 75% to 71% in a generation. Why? Were birds eating the green beetles? Were beetles preferentially mating with the brown beetles rather than with the green? In other words, researchers can literally observe and measure evolution in action while simultaneously investigating the causes of these genetic changes. By understanding evolution on this micro scale, researchers can infer how these same processes have worked over much longer periods of time. For instance, the same changes in gene frequencies, but stretched out over a much longer time, can explain how horse ancestors have gotten larger and have reduced their digits from 5 toes to 3 toes to 1 toe over time. We might call these individual horse fossils different species, though the same natural selection pressures that help explain changes in gene frequencies in beetles can also explain macroevolutionary changes like horse evolution. The only difference is the time scale over which these processes are operating. How do we know that frequencies of genes swimming around in the gene pool have changed? In other words, how do we know that evolution has occurred? One tool that scientists use to know whether something is happening is to line up a series of predictions for what the world should look like if that process is not happening. This is called a null hypothesis. A very useful null hypothesis, called the Hardy-Weinberg equilibrium test, was devised in 1908 by the English mathematician Hardy, shown here at the top, and the German obstetrician Weinberg, shown at the bottom. The basic premise is that if mutation does not occur, and natural selection does not occur, and populations are large, and individuals randomly mate with one another, then the gene frequencies should remain exactly the same from generation to generation. In other words, no evolution. But if the gene frequencies do change, then mutation, natural selection, gene flow, or genetic drift, which are all causes of evolution, must have occurred. Hardy-Weinberg can be difficult for some students to understand. Do not be intimidated by the math. It is quite simple. Let's go through it step by step. First, Hardy-Weinberg used a bunch of letters, P's and Q's. Let's walk through what these actually are. In this table is a simple Punnett square. Imagine that a gene has 2 alleles, big A and little a. The table shows a hypothetical breeding between a heterozygous female and a heterozygous male. But population genetics isn't about individuals, it is about populations. And for that, we need to know how many of those big A's and little a's are actually in the gene pool. We'll use the letter P to designate the frequency of the allele big A and the letter Q for the little a. Frequencies always add up to 1, or 100%. So if the frequency of big A is 70%, or 0.7, meaning P equals 0.7, then the frequency of little a is 30%, or 0.3, meaning Q equals 0.3. If we let everyone interbreed, the frequency of individuals who are homozygous dominant, big A, will be P squared, or 0.7 times 0.7, which is 0.49. So 49% of the offspring will be big A. The frequency of individuals who are homozygous recessive, little a, will be Q squared, or 0.3 times 0.3, which is 0.09. So 9% of the offspring will be little a. The frequency of individuals who are heterozygous will be 2pq, or 2 times 0.7 times 0.3, which is 42%. These frequencies, 49%, 9%, and 42%, all add up to 100%. Let's go through this one more time using slightly different numbers. Suppose the frequency of allele A is 0.6, again meaning P equals 0.6, and the frequency of allele A is 0.4, or Q equals 0.4. The sperm and egg gene pools will be contributing these frequencies to the next generation of offspring. If evolution is not occurring, what might we expect the offspring to look like? Well, the homozygous dominant frequency should be 0.6 times 0.6, or 0.36, also known as 36% of the population. The homozygous recessive frequency should be 0.4 times 0.4, or 0.16, also known as 16% of the population. The heterozygous individuals will be the remaining 48%, or 2 times 0.4 times 0.6, which is 0.48, or 48% of the population. But what if they didn't look like this? What if researchers determine that the population is only 5% big A, and 65% little a, and 30% big A, little a? The only explanation is that the assumptions underlying Hardy-Weinberg have been violated. Remember what the assumptions were? That natural selection, mutation, gene flow, and genetic drift did not occur. In other words, if the gene frequencies are different from those predicted by Hardy-Weinberg, then evolution had to have happened. This is what makes Hardy-Weinberg such a valuable tool for assessing evolutionary change. Let's look in more detail at those mechanisms of evolution, starting with mutation. We discussed mutation in Chapter 2, but that was before we learned about DNA itself. Now that we know a bit more about DNA, let's revisit mutations. The DNA sequence itself codes for a particular array of amino acids that makes a particular protein that makes up a living organism. What if that DNA changes? Then the amino acid sequence might change, resulting in a change of the structure of a protein and a slightly different phenotype. That's exactly what happens with DNA mutations. If these mutations happen in the DNA found in gametes, they can be passed on to the next generation. There are many different kinds of mutations. Shown at the top is a point mutation in which a thymine is replaced by cytosine. As a result of this change, a valine is the amino acid produced. Otherwise, it would have been an isoleucine. Sometimes point mutations result in the production of the very same amino acid and the same protein. These obviously have no impact on the organism's phenotype and are called synonymous point mutations. The example given here, in which a different amino acid is produced, is called a non-synonymous point mutation. This change in the amino acid sequence can have a dramatic effect on the actual protein and what it does for the organism. Another kind of mutation is shown at the bottom. This is called a frame shift mutation. In this case, a nucleotide is inserted into the gene, shifting all the remaining nucleotides down one. Since nucleotides are read in groups of 3, or codons, this dramatically changes the DNA sequence, resulting in entirely different amino acids being coded for, and thus an entirely different protein. Genes can copy themselves and be inserted into different regions of the genome. These are called transposable elements. What causes mutations? In some cases, it is simply a miscopying of the DNA during replication. These are called spontaneous mutations. It is estimated that these can result in one significant new mutation in each individual born. But there are other known causes, such as X-rays and toxic chemicals. They can mutate the DNA, and these are called induced mutations and often have harmful effects on a person's DNA. Another kind of mutation is a duplication. In the case of duplication, whole regions of DNA can be duplicated, meaning that the original gene remains intact and functional, while the copy of the gene is free to accumulate mutations and potentially become a new gene. In the most extreme cases, entire chromosomes can duplicate, resulting in 3 rather than 2 homologous chromosomes. This is called a trisomy, which we discussed in the last chapter in the context of meiosis. Shown here is a karyotype of an individual with Klinefelter syndrome, which is a trisomy that results in an extra X chromosome. Spontaneous mutations can result in all sorts of interesting variation. For instance, a mutation called Leucinium has made the alligator in the upper left white. If this had selective advantages to alligator survival, the frequency of this gene mutation might increase over time, resulting in changes to the alligator population. The cat in the upper right has a mutation that causes it to have white fur and blue eyes. This is a pleiotrophic gene, which also causes deafness. In the bottom right is an example of a genetic mutation that causes this cheetah to have stripes rather than spots. One can imagine how a mutation like this may have been selectively advantageous to the ancestors of striped tigers and increased in frequency over time until all tigers had stripes. A mutation in fruit flies can affect eye development, even producing an eye on the animal's wings. Mutations are the means by which new genetic variants get introduced into a population. It is tempting to think of mutation in a negative way. This is undoubtedly the case some of the time, but mutations are instrumental in introducing the variation that selection can ultimately favor. Take milk digestion, for example. All mammals can digest milk as nursing infants, but adult mammals typically are not milk drinkers, and they can have severe digestive discomfort and diarrhea should they consume milk past nursing age. So why do so many adult humans drink milk? Because of a mutation. A gene on chromosome 2 codes for an enzyme that breaks down lactose, the sugar in milk. A mutation in that gene has kept the gene switched on into adulthood, causing some humans to have the ability to break down lactose long after they have stopped nursing. Who are these humans? Interestingly, geneticists have recently discovered that Northern Europeans and East Africans have a different mutation in their lactose gene, meaning it evolved twice. Keep in mind how natural selection works. The milk does not cause this mutation. Rather, those individuals in the population who did not have this mutation may have experienced severe water loss because they had diarrhea after consuming milk, and may have also been malnourished as a result, and may have died or had less reproductive success. Those with a mutation could digest milk, survive, thrive, and pass their genes on to the next generation. Those human populations that never relied heavily on milk from cattle, for example, some Asian and West African populations, never had strong selective pressures on acting on this lactase persistence allele, and it therefore remains infrequent in the population. Once these new genetic variants are introduced into the population, natural selection can go to work. Over many generations, these genetic variants can increase in frequency and become so finely tuned that an animal as amazing as a leafy sea dragon can evolve. It is time to think about natural selection in more detail. Darwin's ideas of natural selection boil down to 2 things: sex and death. Those individuals who happen to have features that allow them to survive, thus avoiding death, will pass on these features to the next generation through reproduction or sex. By having more offspring, these organisms are said to have a higher fitness. There are quite a few patterns of natural selection illustrated here. These are directional selection, column A, stabilizing selection, column B, disruptive selection, column C, and stasis, or no selection at all, column D. Imagine that we are looking at something simple, such as body size over time in a group of organisms. In column A, those individuals who happen to be small survive better and reproduce more often than those individuals who happen to be large. In other words, small individuals have a higher fitness, as shown in the middle graph. The distribution of size shifts to the left, thus the average size in a population drops, such as in the bottom graph. Stabilizing selection would work against both the very small and very large individuals. Fitness would be the highest in the average size individuals. Because of this, the average body size in the population would not change, even though natural selection was still at work, eliminating the extremes in the population. Disruptive selection is essentially the opposite of this. The average individuals have the lowest fitness, and the smallest and largest individuals are the ones that survive the best. Over time, the average body size in the population may not change overall, but what will result are two clusters, the smallest and the largest, eventually leading, perhaps, to two distinct populations and maybe even two distinct species. Finally, there are situations where a population is in Hardy-Weinberg equilibrium and no selection occurs. Fitness will vary according to other variables, but not size, and the average size will not change over time. Let's look at a famous example of natural selection. In Great Britain, there's a kind of moth called the peppered moth. As the name implies, it is white with spots, and it blends in beautifully with the lichen and bark on trees, thus avoiding constant predation from birds and other animals that eat moths. In 1848, a naturalist discovered a new variety of peppered moth that was completely black. This was described as being of the melanic form, compared to the non-melanic light form of the moth. Though originally rare, by 1950, over 90% of the moths were of this melanic variety. Well, how did this happen? In a word, evolution. During the 1800s, Britain experienced the Industrial Revolution and literally covered the landscape, including the trees, with soot from coal-burning plants. Against a background of lichen and tree bark, the light-colored moth survived quite well. But against a soot-covered backdrop, the light-colored moth was easily noticed and eaten by predators. The frequency of the non-melanic form dropped, but against this dark backdrop, the dark-colored moth was camouflaged and survived long enough to reproduce and pass on the melanic gene. This variety increased in frequency, particularly in the most industrialized areas of Great Britain, like Manchester and Birmingham, as shown in the map in the upper right. Let's look more closely at the genetics of all of this. By treating moth coloration as a single gene with two alleles, we can see in the bottom right graph that the gene frequencies changed over the course of 100 years. The peppered moth experienced quite dramatic directional selection. Now, let's look at this in terms of Hardy-Weinberg. Researchers were able to estimate that during the industrial period, 90% of the moths were of the melanic variety, while 10% were non-melanic. This gives us the phenotype frequencies listed here of 0.9 for melanic and 0.1 for non-melanic. Let's start with the non-melanic. Since non-melanic is homozygous recessive, the frequency of the small C allele must be the square root of 0.1, which is about 0.32. This means that the frequency of the small C allele is 0.32. Since frequencies must add up to 1, the frequency of P, the big C allele, must be 1 minus 0.32 or 0.68. We can then do the math to figure out which moths were homozygous dominant by squaring 0.68 and getting 0.46. That means 46% of the moths were homozygous dominant. 2pq tells us the number of moths that are heterozygous, and since we know P, which is 0.68, and Q, which is 0.32, we can calculate heterozygosity at 0.44, or 44%. So 46% were homozygous dominant, big C, 44% were heterozygous, big C, little c, and 10% were homozygous recessive, little c. This adds up to 100% as it should. Notice that all we needed to do to calculate these gene frequency using Hardy-Weinberg was to observe in nature that 90% of the moths were black and 10% were peppered. But things have changed. Beginning in the 1960s, Great Britain enacted anti-pollution laws, and the trees are no longer covered in soot. No longer is it advantageous to be melanic, and as one might predict, the peppered variety is increasing in frequency. These are the most recent data on the topic. Instead of 90% of the moths being melanic, 90% are non-melanic. Working through the same process we used the last time, the frequency of the small c allele can be calculated as a square root of 0.9, which is 0.95. This means that the frequency of the small c allele has gone from 0.32 in 1950 to 0.95 today. This is a clear violation of Hardy-Weinberg equilibrium, indicating that evolution has occurred. The big C allele has dropped from 0.68 to 0.05, again indicating that the melanic variety is not surviving as well against a backdrop of bark and lichen. But this is not a class on moths. It is a class about the evolutionary history of people. So let's look at an example of how evolution has shaped human biology. There is a gene located on chromosome 11, modeled in the image on the left, known as hemoglobin S, that codes for the production of the protein hemoglobin, which assists with the transport of oxygen around our bodies via red blood cells. However, millions of people have a mutated version of this gene and suffer from sickle cell anemia, which causes misshapen red blood cells, shaped like sickles, as shown in the image on the top right, and serious health consequences. Why would a gene so detrimental to our survival persist in the gene pool? Shouldn't natural selection have eliminated it by now? First, let's look at what causes sickle cell anemia. A very simple base pair point mutation is a difference between those with and without sickle cell anemia. There are hundreds of base pairs in the hemoglobin S gene, but all it takes is just one point mutation from a T to an S, which results in a valine instead of a glutamate in the resulting hemoglobin molecule. This amino acid change results in a slightly different shape in hemoglobin, which causes the red blood cells to be misshapen and causes them to clog up capillaries, cutting off oxygen to vital tissue. Those who produce normal hemoglobin, we'll call that allele A, can have the genotype of either AA or AS. Heterozygotes, therefore, have no complications, even though they carry the sickle cell allele. But the homozygous recessive individuals, SS genotype, have sickle cell anemia, which is fatal for 80% of untreated individuals before they reach reproductive age. This again raises the question, why hasn't selection eliminated the S allele from the population? As you may have guessed from the title of the slide in the previous one, there is another variable to consider, malaria. Malaria is a very dangerous and often fatal disease caused by the transmission of a parasite known as Plasmodium to humans through the bite of an infected mosquito. As shown here, when a human is bitten by a mosquito, the parasite can be introduced into the human host. Once inside humans, the parasite makes its way to the liver and reproduces. The daughter parasites take up residence in red blood cells, multiply, and eventually cause the red blood cells to swell and burst. These little parasites can eventually make their way back into a mosquito that bites a human, where they will reproduce and start the cycle over again. Malaria is extremely dangerous and it kills close to 1 million people every year. Beginning in the 1940s and 50s, researchers began to notice that humans with the faulty S version of the hemoglobin gene tended to live in equatorial Africa, where malaria is particularly common. In addition, heterozygous AS individuals also tended to be the most frequent in these areas. Furthermore, heterozygous individuals tended not to die from malaria as often as individuals who produced normal red blood cells, AA. Notice in this map how the frequency of the S allele corresponds quite well with the presence of malaria in sub-Saharan Africa. It was a young researcher by the name of Anthony Allison who figured this all out. Individuals with normal red blood cells, AA, were susceptible to malaria. Individuals who were homozygous for the faulty hemoglobin gene, SS, often died of sickle cell anemia. but heterozygous AS people could survive both. It turns out that the AS individuals produced red blood cells that do not sickle and are poor hosts for malaria. This results in what is known as a balanced polymorphism, in which the heterozygous individuals are the most fit. But notice what this means. If two surviving heterozygous parents have a child, there is a 25% chance that the child will inherit the S allele from the mother and the S allele from the father and have sickle cell anemia. There's also a 25% chance that the child will inherit the A allele from the mother and the A allele from the father and will thus be particularly susceptible to malaria. But both of these alleles remain in the population since the heterozygous individuals have the highest fitness. The continued work of Frank Livingstone, an anthropologist at the University of Michigan, helped us understand how natural selection has shaped the relationship between sickle cell anemia and malaria. There is evidence that the Bantu carried the A allele, which is the mutated form of the hemoglobin gene, with them when they migrated from Central Africa into South Africa, beginning about 5,000 years ago. But the A allele was not the only thing the Bantu brought with them. They were agriculturalists who cleared forest land for their crops. In doing so, they created large areas where water could pool, the perfect breeding grounds for malaria-bearing mosquitoes. This is an excellent example of how culture helped shape our biology. The introduction of agriculture to this area of Africa increased the prevalence of malaria, which in turn selected for those individuals with one copy of the A allele. Before we leave this topic, let's look at one more aspect of the story. Because hemoglobin production is polygenic, many genes responsible, and the malaria does not do well in any malformed red blood cell, it is perhaps not surprising that other gene mutations have arisen in other areas of the world where malaria is endemic. For instance, there is a disease called thalassemia, which can be found in populations in Asia and around the Mediterranean. This disease affects the synthesis of hemoglobin and can be fatal. But heterozygotes are more resistant to malaria. There is an enzyme called G6PD. The gene for this enzyme is on the X chromosome. A mutation in this gene causes a faulty enzyme that affects the health of red blood cells. Once again, the homozygous recessive individuals can die from a failure to produce this enzyme, whereas heterozygous individuals are more likely to fight off malaria. As expected, high frequencies of this allele can be found in populations where malaria is prevalent, such as Africa and Southeast Asia, as shown on the bottom right map. In Chapter 2, you are introduced to four forces of evolution: mutation, natural selection, genetic drift, and gene flow. We've already gone over mutation and natural selection in more detail. Now let's look at genetic drift. Drift causes a change in gene frequency, not because some individuals are more fit than others, but simply because random fluctuations in frequencies can occur if the conditions are right. Here is an example of this. Imagine that we can model a gene that has two alleles as a simple coin flip. Alleles are the heads and tails. Now, imagine that the population is very small and only two individuals reproduce. Certainly, one individual may give the heads allele and the other the tails allele. In fact, you can figure out with a Punnett square that this will happen 50% of the time. But the other 50% of the time, the offspring will be either all heads or all tails. If the offspring are all heads, the frequency of the tails allele will have reduced to 0. A major change in gene frequency. This is a result of a random nature of coin flips, and not because the tails allele was somehow selected against. As a population increases in size, such as the coin flips increase in number, the likelihood of all the coins landing on the same side is reduced significantly. This can be seen in the top table. As the number of coin tosses increases, the likelihood of drift away from the 1-to-1 expected outcome is reduced. At the bottom of the slide is another way to think of this. Imagine that we have three populations of different sizes. Population A is the smallest, B the next smallest, and C the largest. We can now model how one gene will drift from the starting point of a frequency of 50%. Notice that population A has drifted to the point that its gene frequency is now 100%. Population B has drifted in the other direction, close to 0%. Population C, the largest, has remained the most stable. Humans are very numerous. There are about 7 billion of us, so drift shouldn't have an impact on us, but it can. Some human groups will only reproduce with members outside their group. These are called exogamous societies. Other human societies are endogamous, meaning that they only breed within their groups. If the groups are small enough, they can be subject to genetic drift. For instance, there is a small religious sect in Pennsylvania known as the Dunkers. They are originally founded by a group of 28 Germans in 1719 and have remained relatively endogamous. Compared to other Germans and Americans, their blood types have drifted, with the B and AB phenotypes reducing in frequency and A increasing. Likewise, the MM allele, yet another protein family on blood clots, has increased compared to other German Americans. Because the Dunker population has remained quite isolated, the drifting of these gene frequencies has been primarily by chance. Sometimes a small group of individuals migrates to a new area and becomes reproductively isolated. This is a special kind of genetic drift known as the founder effect. That small founding population probably does not have the same genetic variation as the original, and much larger gene pool, and thus, just by chance, will have a different frequency of certain alleles than the original population. For instance, as we will see in later modules, Native Americans are descended from a small population of individuals who crossed the Bering Strait from Asia about 20,000 years ago. But interestingly, white blood type O exists at very high frequencies in Native American populations. These data, contrasted with robust genetic evidence that Native Americans are most closely related to Northeast Asians, suggest that the small founder population of Native Americans that crossed the Bering Strait had, by chance, a higher frequency of blood type O than the parent population of East Asians. Another example of genetic drift can be found in the frequency of a degenerative genetic malady known as Huntington's disease. Huntington's is a terrible, debilitating disease that causes degeneration of the brain, dementia, and jerky movements of the limbs. It is always fatal. Scientists have learned a lot about Huntington's, including its genetic basis. Everyone has a gene on chromosome 4 that terminates with the sequence CAG valine. In those with Huntington's, the code skips and repeats CAG over and over again, usually 35 times or more. Unfortunately, this allele is dominant, meaning that only one copy of it from either parent leads to Huntington's. Fortunately, the disease is quite rare globally. Oddly though, around Lake Maracaibo, Venezuela, Huntington's is quite common, with half of the villagers suffering from the disease. The geneticist Nancy Wexler researched the origins of this strange clustering of an otherwise rare disease and found that everyone in this community can trace their Huntington's back to a single woman who lived 200 years ago. Because these populations are small, the allele can increase in frequency. And because the symptoms of Huntington's do not materialize until after the age of 40, those with the alleles have already reproduced, passing on this fatal allele to the next generation. As with the Dunkers and blood types in Native Americans, genetic drift is implicated in our understanding of why so many individuals in this community have Huntington's. As we have already seen, for much of human history and certainly today, genes have not been isolated. They flow. We will examine this in the next slide. Gene flow is the fourth and final cause of evolution that we will discuss in this module. Gene flow is also known as admixture. The basic idea here is that genes can flow across population boundaries, introducing new genetic variants into the gene pool. This is illustrated below with a simple figure showing how brunettes and blondes could exchange genes across leaky population boundaries, resulting in a population with a different, more heterogeneous frequency of alleles for hair color. As one might expect, the closer two populations are to one another, the more likely it is that genes can flow. Populations separated by great distances or by great geographic barriers have less opportunity for gene flow. An example of gene flow in human populations is the distribution of blood type B across Eurasia. Notice that in East Asia, type B has a frequency of nearly 30%, but it drops to nearly 0% in far Western Europe. This drop is not precipitous but quite gradual, also known as clinal, suggesting that nearby populations have exchanged genes and that the blood type B allele has slowly flowed east to west across Eurasia over the millennia. Anthropologists have observed that gene flow is highly influenced by social structure. Endogamous societies have little opportunity for gene flow because they do not permit outside individuals into their breeding population. Exogamous societies have higher genetic diversity because of the introduction of new genes into the gene pool. There is another manner in which social structure influences genetic variation. Patrilocal societies are those in which males stay in their place of birth while females migrate out and new females migrate in. One might expect then that in patrilocal societies, the male genetic diversity, as measured by the Y chromosome, would be low, and female genetic diversity, as measured by the mitochondrial DNA, would be high. Those predictions are borne out in a study of 3 patrilocal societies in Thailand. In contrast, matrilocal societies are those in which the females stay in their place of birth and the males migrate. One might expect the opposite pattern of genetic diversity, and sure enough, in matrilocal societies, the mitochondrial DNA has low genetic diversity, and the Y chromosome displays more genetic diversity. One final thought on the process of gene flow is the difference between biological inheritance via genes and cultural inheritance via ideas. Archaeologists have long recognized that the origins of agriculture reside in the Middle East, and that Europeans developed agricultural practices a few thousand years later. But why and how? Did the Middle Eastern agriculturalists expand their population and conquer Europe, essentially replacing both the genes and the previous hunter-gatherer way of life? Or did the Middle Eastern populations expand and gradually intermix both genes and ideas into the European inhabitants? The beauty of modern population genetics is that these hypotheses are testable. What has been revealed in many ways refutes both hypotheses. Modern Europeans can trace their ancestry to well before the origins of agriculture, meaning that the original European hunter-gatherers were not replaced. In addition, there is not a gradient of genetic similarity from west to east that would be expected if gene flow from the Middle East explained the origins of agriculture. Instead, what may be happening is an exchange of ideas in the absence of an exchange of genes. This mechanism of cultural evolution is important to understand as we think about the biology of a cultural animal. Let's review. In this chapter, we've encountered 4 causes of evolution: mutation, natural selection, genetic drift, and gene flow. Mutations introduce new genetic variation into a population. This variation is then subject to the power of natural selection, as we saw in the case of peppered moths and sickle cell anemia. Novel genes can also be introduced into the population from neighbors via gene flow, as can be seen in the distribution of blood types across Eurasia. Finally, small populations can become isolated and experience genetic drift. In the next chapter, we'll examine how these 4 forces of evolution have shaped the pattern of variation seen across human populations today.